everyone. Welcome to another Expert Talk series. I just want to remind you, we bring these to you to help you figure out your challenges, but also we ask you to come to help others here to figure out their challenges, because we are an extended family. And with that, I can only say each one of you are precious. We want to help you, but we want to help you to help everyone here. So always believe in yourself. You are not alone, and you help everyone who feels isolated by coming tonight and sharing just your presence. So thank you so much for being here. And with that, I'll turn it over to our CEO and founder, Indu Navar. Welcome, Indu.
Thank you, McFinn. And, I would love to start today with welcoming Dan Fowler and Brian Radecki, whom we are honored to have today. And we're going to share a lot about RAPA Therapeutics. But before we go there, I would love to share with you, many of you know Martha Beach here, and she was, featured in the May ALS Awareness Month by the television in their area with the Tampa. So this was a 3-minute video I'm going to just play for everybody as a kickoff to, May as a month. So this is Martha and Paul Beach story.
Welcome back to Spectrum Bay News 9. In your health news, a nonprofit is using AI to analyze audio and video recordings of ALS patients. The founder of Everything ALS lost her husband to the neurological disorder that impacts nerve cells in the spinal cord and brain.
In his honor, she works to bridge the gap between patients, research, and technology.
We are focusing on both prognostic markers, so we can actually bring good measurements during clinical trials, and also what we call diagnostic markers that can be used for early diagnosis.
Spectrum Bay News 9's Melissa Eichmann introduces us to a couple enrolled in the speech study.
Can I say, oh, great.
Paul Beach is part of a speech study to help advance the diagnosis and prognosis of ALS.
Count up from 1 for as long as you can until you run out of breath. Oh. Great.
Paul, a successful businessman and active weekend warrior, was diagnosed with ALS in 2019.
He was having a hard time buttoning his shirts and his ties, a little bit of difficulty writing, and then felt clumsy.
His wife Martha's voice is also recorded as part of the research. He, the advanced registered nurse practitioner in the area of neuroscience, describes Paul's progression, saying he developed symptoms with speech over the last 6 months.
Initially, he had very little symptoms, as most people do, and the concern for us was how progressive, how rapid, which people are unable to predict.
Using an assistive device, Paul says he hopes participating in this study will help change those predictions.
With the technology available now, precious time can be saved. I want people to be able to be diagnosed earlier.
The couple is also concentrating on helping other families through that diagnosis by launching the ALS International Foundation.
We are focused on care. One of the areas, the opportunity that we see is just helping hope, psychological counseling, and that ability to help everyone that's been affected emotionally and psychologically.
And Paul is writing a book, a personal perspective on how words have mattered.
You can give someone hope, or you can take it away from them very quickly in the way they say those words.
The Beaches hope getting their message out in several ways helps raise awareness.
So much of our lives we take for granted. Simple pleasures like a glass of wine or touching your lover's face. I want everyone to relish these things.
Sharing their personal journey to empower those impacted by ALS.
He's my hero. He gives me strength. I couldn't do this without him.
Strength and hope to help others, no matter how it's voiced. In Pinellas County, Melissa Eichmann, Spectrum News.
To learn more about the Everything ALS speech study, head to our website, baynews9.com.
Join Spectrum Bay News 9 tonight at 9 o'clock for the state of mental health in America. Dr. Nicole Cross and
Thank you. And, this is what makes difference when we come as citizens. And today I would love to welcome, both Dan Fowler and Brian Radecki, who are from RAPA Therapeutics. Dan Fowler in 2017, Dr. Fowler became founding chief medical officer and chief scientific officer at RAPA Therapeutics, building a team of scientists and research lab, and eventually a GMP manufacturing facility from scratch. His overarching vision at RAPA has been to extend the bench to bedside paradigm developed during his NIH career to his full commercial application. This approach uses epigenetically programmed next generation RAPA T cells for a curative therapy of refractory cancer and elevating the devastating neurodegenerative diseases such as ALS. And as we'll hear more, and Mr. Brian Radecki is the founder, CEO, and member of the board of RAPA Therapeutics, a clinical stage startup biotechnology company. And Brian co-founded RAPA after being introduced to Dan Fowler by his business partner, Bill Kapner, and learning about the exciting cell therapy technologies he has developed during his time at NIH, which was revolutionized treatments for cancer, autoimmune, inflammatory, and neurodegenerative disease. I have had real privilege of having met with Bill Kapner, and who was diagnosed with ALS. And so today is a very special day because RAPA Therapeutics was really founded by somebody, like us, who actually was affected by the disease. And Dan and Brian came together with Bill to found RAPA Therapeutics. So we today, credit Bill for what he did, even though he's not here with us. We think about him every day, and he's helping touch all of us in many different ways. So with that, Brian and Dan, thank you, and the floor is yours.
Thank you, Indu. I did not know you were going to say that, and I really do appreciate it because Bill is in our thoughts every day. So I appreciate you mentioning Bill. Hi, everyone. My name is Brian Radecki. I'm the CEO of RAPA Therapeutics. As Indu mentioned, Dan and I co-founded this together. I always say we're a very good team. He's the science. I'm the business and operations person. And so far over the past 6 years, we've been able to do some amazing work, which we'll talk about. I'll just give a little intro in the first few slides to try to get Dan really the floor to really describe the cell therapy program that we have here, specifically the expanded access program that we're working with Mass General and the Healy Center. Let me start by first thanking Everything ALS. Indu, you and your team have been amazing. The tireless work that you do, we really appreciate that. We've obviously had a relationship a few years ago, and now that we have this expanded access program, it's great to really be sort of joining your organization. Dan will talk a little bit about Everything ALS is also a part of our trial, and they will be doing a lot of at-home monitoring and other things. And so Dan will talk a little bit about AI. I heard that earlier on the call. We're also using AI on this trial. So this is one of the most cutting-edge trials that's out there. I happen to be talking to Merit Sokovich earlier today, and that was the conversation. Also, I would really like to thank all the people living with ALS and their families for working with us and all of us on these trials, as we really aim to slow, stop, or reverse this terrible disease. Really, you are all doing the hardest work that's out there today. So just to give everybody a little timeline, I'll tell a little story, which Indu sort of mentioned. Dr. Fowler had been working at NIH for almost over 27 years, working on various cell therapies, on cancer programs, on neurodegenerative programs. He spent a good amount of that time working on T regulatory cells, which he will talk a little bit about later. But really, everything was surrounding the cell therapies, really utilizing the body's own immune system to really help cure it from a lot of these diseases. This is a very, cutting edge work. Cell therapy has really come to the forefront in the cancer side the past 5 years, and really is just sort of getting over to the neurodegenerative side. There were some early pioneers like Stanapel, who had been, talking about Tregs, but as I mentioned earlier before, some people maybe got on the call. We're just honored to take the baton and run this leg of the race as best as we can. While Dan was working at NIH, I spent 25 years working at different public companies, almost 20 years at a commercial real estate information company. I was one of the early employees and ultimately retired from there as the VP of Operations and Chief Financial Officer. And when I retired, my good friend Bill Kapner and I, who was an entrepreneur himself, he had several businesses, started working together. I had invested in one of his companies a decade earlier, which was very successful. And through that process, he invited me to lunch to meet this Dr. Fowler that he knew. And that was in 2016. And it was by the end of that lunch, all three of us looked at each other and we said, okay, let's start a company. Like, let's really try to, spin out Dan's life work out of NIH and really try to get it from the bench to the bedside. And so, I went home that night, talked to my wife. She told me I was crazy. I was a commercial real estate and finance guy. What was I doing going into biotech? And I said, don't worry, I'm not running the experiments. We got the expert here from NIH. So, but the next day, I think I got the name and incorporated in Delaware, RAPA Therapeutics LLC. In the first 6 months, the company was at my kitchen table, basically. Dan then had a lot of work to do. He had to finish up a lot of the trials that he had going on at NIH, and he retired from NIH in August of 2017 on a Friday, and on a Monday started at RAPA Therapeutics. I mean, obviously co-founding it with me. This was a true startup. I mean, it was Dan and I, we went around, looked for warehouse space. I mean, we can't tell you some of the places that we saw. We ultimately found a warehouse near NIH, right across from NIAID and near FDA in Rockville, Maryland, and we leased it out and built out a amazing research and development lab for Dr. Fowler, and spent the first 2 to 3 years, basically sort of rewriting the platform. Dan ran tons of IND enabling experiments and mouse models and different things, and really created the platform, two products, a cancer product and a neurodegenerative product, which we're first looking at in ALS. And then after that, first 2 to 3 years, we had a very important decision in biotech, especially in cell therapy. If you read about it, was to either quote unquote outsource our manufacturing cell therapy or build it out ourselves. I think 90+% of people, especially small companies, outsource everything. We decided to invest in ourselves, invest in the company more, Bill and I, and sort of build out our own GMP manufacturing. So we spent the next year working with FDA, doing Type C meetings, and essentially designing and building out right next to our R&D lab a fully GMP approved manufacturing center, which I think now we've already had like 75 products we've gone through. We can do 300 or 400 a year, which will satisfy the current trials that we have. And obviously, as we hope to move to commercial, then we will have to look to expand even more. So that kind of leads us up to the last 2 or 3 years where, after we've done all this work, we, Dan went to FDA with an IND and protocols, one in cancer and one in ALS, which were approved. And we just finished those trials, those initial phase 1, 2 trials, very successfully, proving our products are safe and with really, really good signals of efficacy in all these diseases. So now we're sort of moving into this next phase, which will be sort of phase 2, 3 trials with much larger patient populations, more centers that we'll be treating at. And so that's sort of the current phase that we're in. This is just a couple things on the center. I was actually going to do a video, but I wasn't as technologically advanced as you, but it's really a great center. If you're ever in Washington, D.C., we love to give tours. We give tours to people all the time. The staff love it when people come through, but it's, right near NIH, FDA, we're in between three major airports. Again, this is all dedicated for our products, and we can basically support, we built it sort of commercial ready so we can support all the way up to commercial trials. So it's really an amazing center. And, through this journey, we've obviously been fortunate enough to work with many great organizations. Like I said, we had met Indu a few years ago. And now, I mean, like I said, we couldn't be more excited on this expanded access trial to be working with them for at-home monitoring, also working with Origin Data Sciences on an AI cohort. And of course, can't thank, Mass General, the Healy Center enough. James Berry is our lead doctor, and we work a lot with Merritt and Suma and a lot of the other doctors there. So we're very fortunate to have, a great team of people and organizations that are working on there. As Indu mentioned, one of my closest friends, Bill Kapner, who sort of really started it all. I'm not sure I would have, when we were at that lunch, he said, let's start a company. And then he looked at me and he's like, you're the business guy, you're running it. And of course, Dan was the science. So I couldn't thank Bill enough. Unfortunately, Bill's life was cut short by ALS. Dan and I went through that entire process with him. And so, I know firsthand, from the day he learned, up until when he wasn't eligible for any trials, I understand what he went through. So what was important to me was to not only move into these phase 2, 3 trials, but also to create an expanded access program where we could treat patients that really can't be treated on any other trial, believe it or not. I mean, I told my wife that, and she's like, I don't understand. I thought that's what experimental trials are for. And I'm like, well, there's actually still inclusion, exclusion criteria for even the experimental trials. And so that's what we did. We partnered with the Healy Center and James Berry. We applied for an NIH grant. And, we did all this work last year as we were finishing up our trials. We obviously submitted, all of our data to them, all of our, the promising data that we had. And essentially we got this grant, it's a 3-year grant, to run this expanded access program to get more RAPA therapeutics out to patients. And these are all essentially for patients that really can't get on any other trials. And Dan will walk through the details of that. So we're just very fortunate. This was very important to me personally. I mean, I'm doing this pretty much, because I really want to make a difference. And I know this, like I said, with my friend Bill. And, I couldn't be more fortunate to be working with Dr. Fowler, who I'm going to turn it over to in a minute. He is really, on the cutting edge. He has been for over 30 years, and the work that he's done and the hundreds and hundreds of patients that he's treated. Every time I go to his house on the holidays, there's all these baskets and gifts from all these different people and artwork and other things. You wouldn't believe, how much people really appreciate his work. But I just, again, I'm grateful that I could be working side by side with him, doing my part. And with that, Dan, I'll turn it over to you to talk a little bit more about the expanded access program.
Thank you, Brian. Maybe you can go back to one slide. So you may, if you're looking at my background, you'll see I have a statue of the Rapa Nui statues that are very prominent on Easter Island. I'm not down there right now. This is actually a replica that's in my backyard. You can also see there's a little snow on there. So that was taken last winter, but one of our friends and investors got that as a gift for me. It sits right out my kitchen window. So whenever I'm cooking or doing the dishes, I look out at that, and then I know I'm supposed to be working on the RAPA. But it does mean a lot to us. It means a lot to be here today talking about our expanded access protocol, and I'll lead you through that. As Brian's saying, we're very fortunate to be working with MGH. We were just up there recently, meeting with Dr. Berry. Also, Suma and Sabrina are involved in this EAP, which is very important to get many different experts into the design of this project. If you see at the bottom of this slide, for anybody that wants more detailed information on the EAP protocol, you can see it there on the clinicaltrial.gov website with that protocol identification number. Next slide, Brian. This slide shows the definitions. Brian touched on it. What does that mean, expanded access? According to the NIH neurology definition, in terms of the grant that we were very fortunate to receive, the protocol should involve the treatment of individuals who are not eligible for other clinical protocols. And that means basically all other clinical protocols. So we had a challenge to how are we going to design that? And what we decided on was for individuals, patients living with ALS who had a slow vital capacity of less than 50% of the predicted normal value. And if you look on clinicaltrial.gov and you do a search, you basically won't find any treatment protocols where such patients would be eligible for investigational treatments. So that was our definition. I think a very stringent definition. The other thing to point out about this EAP, which really worked to our advantage in obtaining the grant, was that it had a very scientific angle to it. In other words, the idea is not simply to get these cells out and give them to patients and then not get return on the understanding of how these cells may or may not be working. But the EAP protocol is loaded up with big scientific objectives. Number one, to look for the safety of these cells, but also to look for potential signs of clinical effects. And then loaded up with all the immunology, all the science, all of the protocol design issues, such as the AI-driven, so-called synthetic control work that we're doing with Dave Ennis over at Origent. So there is no real placebo control or any other control on this protocol. Every enrolled patient is intended to receive the RAPA 501, but we're using this synthetic control to try to obtain some better understanding of the potential benefits of the therapy. And then the other key thing is with everything ALS, we've already gotten started on our first couple patients. Stephanie over there doing a great job leading the team proctoring. So everything ALS will actually go in and meet these patients virtually. And we're already setting them up for the remote monitoring, which is very critical, especially in this subset of patients, as oftentimes you lose a lot of data because the patient may not be healthy enough to get into the hospital, to the university. So that's a key part of this whole effort, is to try to capture as much data as possible to try to help the patients and help the scientific understanding of the research. I'm sorry that was a long discussion, but I think it is important to understand what is the definition of the EAP. In terms of other things that we decided to do on this protocol, in addition to the slow vital capacity, we opened it up to pretty much everything else that some of these are oftentimes restrictive on clinical protocols in terms of whether the disease is sporadic or familial, the level of L-Escorial criteria, whether people can continue other medications, or whether how long the disease has been established in a particular patient. So we took off the restrictions on all of these to try to open it up to as many individuals as possible. I will note the one thing there at the end, because this is a unique immune T cell therapy where we have to collect the T cells from each patient, we set in a restriction that patients have to have a pretty good CD3 positive T cell count to get on the study. Even there, though, we set the count at 500. Most individuals will have a count around 1,500 or higher, but we did have to put on some lower limit to help make sure that we could actually manufacture the product. Next slide, Brian. This is a cartoon. If you go to the showing basically the overall process here, if you go to the upper left quadrant, you'll see step 1 in the RAPA process, where patients' cells are collected in a blood bank. For this protocol, they're collected at the university blood bank. It's about a 3 or 4 hour apheresis procedure. If you know anybody who's donated platelets and so forth, very similar process. Those cells are then shipped to RAPA, and then we receive them usually on the same day. And then on step 2, we take those cells and we bring them into our GMP manufacturing facility in Rockville, Maryland, the one that Brian was talking about, that we build from scratch very successfully. It's a finely tuned manufacturing. We have a great workforce that is able to manufacture these products. And so the next morning that we receive this product from the patient, we clean it up. By cleaning up, I mean we wash the cells, we purify a little bit the immune system, and then we put it into culture in incubators. And we do that for about a week. And while we're doing that, we add our proprietary mix of cocktail, we call it, that reprograms the T cells. As you likely have heard in ALS, a big part of the problem is neuroinflammation. It's only one part of the disease, but it's thought to be a very important part, where the inflammation of the patient's immune system will aggravate the degeneration of the underlying ALS process. So what we're doing basically is taking the immune cells that are have an inflammatory profile, and we have to deprogram them. And we do that not through genetic engineering, but we call it epigenetics, where we're using different molecules to de-differentiate those cells so they kind of lose their memory. And then in step 3 in the lower right quadrant, we then redifferentiate those cells. We give them new instructions to become an anti-inflammatory T cell. And that occurs, the total process is about a week. We then cryopreserve those cells at our facility into single infusion aliquots that are then sent back in step 4 in the lower left. Those cells are then shipped back in liquid nitrogen. So they're at a very deep frozen state and very stable there for many years. And those cells are shipped back to the doctors at the university, who can then thaw them out at the bedside. Usually, almost always, actually 100% of the time in our case, done as an outpatient. So this is not an inpatient procedure. It's done where the cells are thawed, they're infused in through an IV, and then the patient goes home, about a half a day procedure as an outpatient. Those cells then engraft into the body and are quote unquote living drug, where they'll circulate through the body, hopefully reestablish the immune system to be more anti-inflammatory. Next slide, Brian. This is a little bit of a complicated and overly scientific description, but I know there are some scientists in the audience today. I know actually, Indu, you're pursuing your own scientific career, so this one's for you. That's a little bit of a joke. But if you just bear with me for a minute, I'll go through some of the details of this. If anyone has any questions on this, I'm certainly available at future dates to discuss. But in the upper left, number 1 is a motor neuron, and that's thought to be the initiating event where the neuron degenerates. And if that's not bad enough, the degeneration of the neuron will cause a lot of inflammation in the brain. And then you see in the lower part of the diagram, around the numbers 4 and 5, you see pathogenic Th1 cells. So T cells exist in different flavors. The Th1 flavor is a very aggressive flavor of T cell that will kill targets and cause a lot of inflammation. So what happens is these pathogenic Th1 cells will make the disease worse because they go into the brain and they see the inflammation from the degeneration, and they actually exacerbate and propel the course of disease. As Brian mentioned, a lot of this biology described by Dr. Stan Appel. We were very fortunate to meet him and get a lot of advice from Stan, who helped describe a lot of these interactions in ALS, first in mice and then in patients. And we'll describe a little bit how our cells are a little different in terms of how to get a handle on this kind of very complicated neuroinflammation. Well, what we have here with the RAPA 501 in the upper right is that little blue cell is the RAPA 501, which is a form of Treg cell. We call it an iTreg. I means inducible, and I'll describe that a little bit. So it's not a natural Treg, it's a reprogramming, as I mentioned, an epigenetic reprogramming. So it's an iTreg, and it also has a Th2 flavor. So T cells exist in these different flavors. I've done a lot of work on Th2 cells. So what we decided to do is make our RAPA 501 both Treg in characteristic and also Th2. So it has a bimodal mechanism of action, which we believe will make it a more reliable cell to tone down the neuroinflammation. And so the idea here is in a particular patient, we add in the blue cells. The blue cells will cross-regulate, down-regulate the inflammatory pathogenic Th1 cells, and then we hypothesize that we will see an effect where there's not as much damage to the central nervous system. Next slide. As I mentioned, this is another cartoon that gets a little bit into the immunology of all of these different populations of cells that are thought to exist in the T cells of our bodies. On the left is the thymus. The thymus is that small organ that sits above the heart, and the thymus is the source of T cells as one is born. And the problem is the thymus involutes with age during adolescence. So by the time individuals are adults, there's very little new T cell production, which is one of the problems with aging, contributing to that. Now on the far left is the naturally occurring Treg cell. This would be the cell that Dr. Appel and many others focus on for Treg therapy. You're basically trying to find those natural Tregs that have come out of the thymus. They haven't been activated yet, and you're trying to put them into culture and expand those up into bigger numbers and reinfuse those back into patients. Well, what we have with the RAPA 501 is something totally different. On the right, we have actually a process that will take cells that have already been through the T cell activation process in the body. They're so-called post-thymic. They've already been out there. They've been educated. Maybe they're inflammatory. They've seen different stimuli for the immune system. And that's where we put those cells through that dedifferentiation procedure, where we cause the cells to lose some of their memory. And we do that through a rapamycin type process. So I'll get into that a little bit, but rapamycin is a drug discovered on Easter Island, as I mentioned, that binds to and inhibits a molecule called mTOR. Now it gets a little more complicated. There's both a mTORC1 and an mTORC2. That's a whole nother discussion. And when we were at NIH, we were using rapamycin to accomplish this. And at the company, as Brian says, we did a lot of IND enabling research and looked at a lot of other ways. And we believe we've picked up some information that allows us to even more powerfully inhibit the mTOR pathway, which allows us to erase the memory to a more stringent level. And then, as I mentioned, when we redifferentiate the cells, we dial in both a Treg and a Th2, the so-called hybrid phenotype. So you can see how our cells quite a bit different from what you might have read otherwise on Treg. Next slide. This little cartoon, again, if you go to the upper left, it's a little lollipop diagram here with different colors. I hope you enjoy the color, all the different colors. If you get tired of looking at the science, but I'll briefly go through this. Bear with me. In the upper left, you see that when we block mTOR, and again, we have to block both mTORC1 and mTORC2, or you can't grow a good Treg cell in our experience. So you have to be very stringent and basically turn mTOR off. And then in the upper right, what happens as you turn mTOR off, the cells go through a process of autophagy, which you may have heard about in the popular literature as an anti-aging strategy. And what also happens with autophagy is the mitochondria undergo autophagy. Mitochondria are the powerhouses of cells. They also control whether a cell lives or dies. They control the so-called apoptotic tendency. And we know with our RAPA process that we change out the mitochondria, we get better mitochondria that are more fit for performing different functions, and they also create a more durable T cell that doesn't curl up and die when they first go back into the body. And then in the lower right is the memory discussion. So I mentioned we erase some of the memory, and what we get is a cell that's more rejuvenated. It's more of a central or stem memory. It's a little more primitive in its memory profile. And these cells also have very low immune checkpoints. Now, you may have heard about immune checkpoints if you're watching the news channel or the golf channel. You'll hear about Keytruda, which are the checkpoint inhibitors for cancer therapy. So the immune checkpoints are a huge part of immunology research. What we're trying to do here is the opposite. We're trying to eliminate all the checkpoints so that when we inject our cells back into the patient, they don't get immediately turned off. They can stay on and do not get immunosuppressed themselves. We want these cells to be the dominant cell in the body, because if you look at the lower left, these in the light blue, these cells have an immune suppressive function, which is a very multifaceted way in which they can suppress the inflammation. And so we have a cell that's a little bit complicated if you look at the mechanism of action, but we've studied this now for a couple decades, and it's seems to be we're always interested in increasing our understanding of these cells, but it's very fascinating. Now, if you go to the next slide, these are actually some of the experiments performed on our patients living with ALS who have undergone our phase 1, phase 2 trial. If you look on the left, 2 panels is the Treg. So Tregs are defined in part by a master transcription factor, a molecule that dictates thousands of other proteins, and that's called FOXP3. So if you have a cell that expresses FOXP3, it's a good candidate for being a Treg cell. And as you can see, this particular patient at the start of the culture only had about 1.1% FOXP3, but at the end, our RAPA 501 up to 37.3%. So a massive upregulation in this master transcription factor. And then on the right is the hybrid Th2, which are defined by a different master regulator, that's GATA3. And you can see at the start of the culture, 0.1% in this particular ALS patient. And then at the end of culture, the product goes back into the patient, 33.5%. So again, a massive upregulation. And this is how we define these cells. And in fact, we have our own QA, QC laboratory at RAPA and our own staff to do these experiments. And if the cell does not express certain increased levels of these 2 key molecules, then the cells are not suitable for administration according to our definition and according to our FDA filings. Now, I will say every product has passed, so it's worked in 100% of the time, and now we've done more than about 15 manufacturings. And if you look at the next slide that Brian just pulled up, we should talk a little bit about stem cell therapy. So you can see in the left panel, this is measuring CD150. Now you might ask, well, what the heck is CD150? It's a marker on cells that helps define how primitive they are in terms of the stem cell biology. So a stem cell is a replicating cell. It's self-replicating. It can give rise to a lot of progeny, and it won't exhaust itself because it can keep making new cells. That's one of the definition of what a stem cell is. And as you can see, our RAPA 501 in this particular donor, massive upregulation of CD150, consistent with a T stem cell phenotype. Now that needs to be differentiated from other forms of quote unquote stem cell therapy, which you may have read about and heard about, especially in the ALS clinical trial arena. There's the neurologic stem cell. So you take a primitive cell and then can differentiate that cell into various neurologic cellular types. And there have been various trials on that. And then in the middle, there's hematopoietic stem cell transplant. You probably know individuals who've gone through bone marrow transplant or find a match for various blood cancers. So brother or sister is the donor, for example. That's a hematopoietic stem cell therapy. And then off on the right is the immune T cell, T stem cell, which we believe that the RAPA 501 fits into that category. Next slide. Again, this is getting a little bit into the weeds, but I wanted to show a little bit more about the testing and research that we do on the RAPA 501. If you go to the left figure there, it's a little cylinder that shows how we can measure whether our RAPA 501 from a given patient is a good suppressor cell. In this cylinder that we put in the incubator, the well that we place the cells into has a membrane that separates the RAPA 501 on the bottom, that's kind of the clear color, from the color on the top is the orange. So these two different cells can talk to each other, but they can't touch each other because they're separated physically by a membrane. And on the top, we put in inflammatory T cells. We cause a T cell to be inflammatory. And then we add in our RAPA 501 to the bottom, and we ask the question, can the RAPA 501 communicate with the inflammatory T cells and change them for the benefit? In other words, tone them down to be less inflammatory. And as you can see on the right two data, in this particular experiment, indeed, the RAPA 501 is able to communicate with the T cells and tone them down, as evidenced on the far right panel, where now the T cells start to express one of those immune checkpoint receptors, the PD-1 that I was mentioning. So when an immune cell starts to express PD-1, it's going to become quiescent and basically senescent and non-inflammatory anymore. Next slide, Brian. And then we adapted the assay to look for microglial cells. If you're following some of the literature, there are other cells in the brain, the microglial cells, which are thought to be one of the earliest cells that become inflammatory. These are resident in the brain and really help control whether the brain is becoming inflammatory or can be toned down to be anti-inflammatory. There's different subsets of these CNS microglial cells. So we obtained a CNS microglial cell line. It's a human cell line that was discovered and now become available. And now instead of putting inflammatory T cells in the top, we put in inflammatory CNS microglial cells. And then we ask the question, can the RAPA 501 communicate with those cells and tone them down? And as you can see in the right, when we measure the inflammatory cytokines that are made from these microglial cells, you can see that the RAPA 501 indeed can start to tone down some of the inflammation from these microglial cells. Next slide, Brian. Yeah. So that's all nice. That describes the product, but what about in patients? As Brian mentioned, we have completed the phase 1 study. The RAPA 501 was extremely safe. We didn't see any adverse events attributable to the RAPA 501. When I say no adverse events, not of any grade, not of any type. So it was a very apparently safe treatment. And then the question is, well, can it actually tone down the inflammation? And this is an example of one of the molecules that causes inflammation in the body. It's called MDC. It's actually a chemokine. And you can see in one of the patients that we treated on the left, it's on a logarithmic scale. If you look at the y-axis, it's a logarithmic scale. And you can see that there's a massive downregulation in this patient of this inflammatory molecule. And then you can see on the right panel, we asked, well, what about other patients? We have lined up 6 different patients here, and you can see it was a pretty consistent effect when we looked across. And this is the kind of research that we're doing on the phase 1 study, and we're really looking forward in the EAP to confirm and extend these type of biomarker studies. Then the question is, well, what are we doing about the disease? Are we helping to help make people breathe better? Can we slow down the ALS FRSR score decline? These are all things that are very difficult to do, obviously, in a phase 1 study where you're only looking at 9 patients. We're now up to 12 patients with increasing accrual to that initial study. So you're not really well suited, and that's why the EAP with 40 patients is going to give us a little more power to answer that question. But indeed, we did see some trend toward preservation of pulmonary function. If you line up some of the patients that we treated at the higher doses of RAPA 501, that's in column H there on the far right. Over a period of several months, we did not observe the standard decline in slow vital capacity that one sees. If you look in the literature at various studies, reference values A through G, I put here. A lot of this work summarized by Jinsey Andrews over at Columbia. Very beautiful paper where you can do the math, and basically the slow vital capacity will decrease by about 3 to 4% every month, almost by clockwork. Obviously, there's some variability to that. Some patients not so much. But in general, a lot of the math has already been done, and all of the intervention studies really have not been able to make any impact on slowing down this decline. So with these preliminary data, we hope that we might be seeing a signal here. And that's another very compelling aspect of this EAP trial, the fact that we're going to be treating people who are already down below 50%. It'll give us a more stringent readout to see if this is a real effect.
Dan was being nice and labeled it A, B, C, D, E, F, but that's basically trials, and that's basically approved or former approved drugs, all that have been out there. So, without putting any names on there, you can see a big difference in the stabilization of the slow vital capacity, what we saw while people were taking the RAPA 501 versus anything that's been approved ever. So just want to point that out. Dan, again, Dan's being nice on.
Yeah, thanks, Brian. Trying to be nice. Next slide, thank you. So let's get into, just in the last few minutes here, some of the nuts and bolts of this EAP study, which we're very fortunate to be able to do. We're just now getting started. I'll talk a little bit about that. We have a jump start at Mass General Hospital because of our experience there. So we've already have 2 patients on the study at Mass General, 2 individuals, and we've already manufactured the product. We're shipping the product out to the first patient this week. They're going to get the infusion next week. Second patient already manufactured. QA, QC look great. We're sending that out. And we have 2 more patients that James Berry and his team are screening over the next several weeks. So we're off to a pretty fast start. Now, the goal is to get this started at 10 different sites across the country. A couple reasons for that: we want to increase the investigator experience with the rapid treatment, and also to try to get a geographically and ethnically diverse patient population. You can see some of the centers here. Each of these centers is in different stages of working through the IRB process and so forth, but this gives you a little idea of the scope of the project.
And bear with us. I do see some questions in the chat. We'll try to get through all those when we're done.
Yep.
Just to throw it out there. So our goal is to have all these centers up and running, by the end of the year, and some of them will be up and running in the next few months. And then, we do want to continue to expand centers. So we will look at other centers next year. We'll also be starting additional trials. And so we'll be using the same sort of network and the Niels network. I was actually literally just talking to Merritt about this a few hours ago. So, again, this is a process, but we'll be excited to get it out to as many centers as possible. And just to kind of put that there. Go ahead, Dan.
So Brian and Dan, we will be moderating the questions. So after this session, we'll take care of all the questions.
Okay. Okay, great. Thanks.
So I mentioned our plan is to treat 40 patients. The primary objective is to make sure it's safe and feasible. And then we have a lot of other objectives, looking at the disease activity scores, looking at the immune system, markers of neurodegeneration, effect on pulmonary, slow vital capacity. And then, as we mentioned, it's beyond the scope of this talk to get too much into it, but Dave Ennis over at Origent has developed beautiful algorithms for doing so-called synthetic control groups. So we don't use a placebo control. We're trying to work with Dave to work with existing data out there in the literature to determine whether the treatment might be effective. And then the other objective, I'll say the most exciting one, is to work with Indu and team at Everything ALS to do the remote monitoring. Next slide, Brian. So what about this collaboration with Everything ALS? The first aim is to, with respect to accrual, we want to help ensure prompt accrual. We want as diverse a patient population as possible. As you know, Everything ALS is the largest ALS citizen community that's focused on diversity and inclusion, has more than 1,000 research participants across every state, and has maintained ethnic diversity across their participation in these studies. Aim 2 is the remote monitoring, which we're off to a great start. We already have the first 2 patients getting started on the remote monitoring. The ALS, Everything ALS proctors help these participants with this remote data collection. A cell phone is provided to the patient that's installed with the Everything ALS app. The sessions are performed about every 2 weeks. And these testing is intended to supplement the on-site study testing. So it's meant to augment what we're doing at the university, which is about an every 42-day testing. However, if a particular patient is not able to make it, we have this as the sole backup for data collection. And what do we do in the remote testing? We do the surveys, we do pulmonary testing, speech analysis, and the protocol will also look at activity monitoring. So a fairly ambitious amount of data collection, and we're looking forward to help get that implemented. And the last slide, a special thanks to all the clinical trial participants and families. Special thanks to the NIH, Dr. Barry and his entire team at Mass General, in particular Megan, who we met with yesterday, is doing a great job of study startup and getting patients onto this study. The entire team at RAPA, in particular Jenny and Sylvia, who've been instrumental in getting this study going and will be there to help it come to fruition. It's a big task, but we're up to the task. We have a great CRO, Osmosis, who's keeping us on schedule. We are having a great success working with these 10 different clinical trial sites. There's a lot of bureaucracy, but we're breaking through it and making great progress on getting these sites up and running. All of the investigators are very motivated. That's certainly not any rate limiting steps there. Working with Dave Ennis at Origent, Everything ALS, we'd like to thank the ALS Association was instrumental in some of our early work with the biomarkers, and also shout out to ALS Northwest, which was instrumental in some of our funding. I'll stop there, Brian.
Thank you, Dan.
Thank you, Dan. That was great.
Perfect. Well, thank you for giving us an excellent overview. And we have, as you've seen, a lot of questions. So Martha Beach and I will be asking those questions. So we'll spare you from going to the chat. So
Thank
we'll
you.
be collecting. Thank you. So the first question is RAPA 501 expanded access protocol or clinical trial recommended for bulbar patients? If yes, what should I do to enroll in it considering I'm in the West Coast? And is there a cost to the therapy?
Yeah, I can answer that. So there's no discrimination against a bulbar. It doesn't count against eligibility. The main thing would be it has to be the slow vital capacity under 50%. So that's once a particular candidate qualifies under that criteria, then everything else kind of flows from there. So would not expect any exclusion based on any other particular situation. In terms of the second part of that question, on the West Coast, obviously MGH is the furthest thing from the West Coast. It's a beautiful place and all that, but patients on the West Coast deserve to be treated close to their locality. That's why we're trying to get it set up at many different centers on the West Coast. Mayo Clinic, Arizona, Barrow Clinic, UC Irvine, UCSF, and Providence Hospital at Portland, Oregon. These are all 5 centers that have all been working diligently to try to get this started. So a particular patient could contact me and we'll try to work with them to direct them to whatever site. It's always hard to predict which site is going to be able to break through the bureaucracy first. If I speculate, I would say in the next month or two, hopefully we'll have one of the West Coast sites up and running, but I can't make any promises on that other than we're going to work hard every day to get that to be a possibility.
Just to add to that, so there isn't any cost to the patient. Again, thanks and shout out to NIH for funding this program over the next 3 years. So it will be just whatever the patient has for their own travel costs. And then as Dan said, this is another big step for our company. We're ready to go. So, I mean, getting all these centers up, we have the CRO helping and it's really getting through, as Dan said, the bureaucracy at each of the centers. But this is going to be a launching pad because then we have a few more trials that we will be starting next year and the following year, which would be even larger trials. And we'll use all these places as a launching pad. As at MGH, we're able to immediately start accrual. We'll be infusing our first patient, as Dan mentioned, next Friday. The following Friday, another patient. He's got 2 other patients in the hopper. So we're very excited to, have an expanded access compassionate use program to get out to as many people as possible.
Thank you, Dr. Fowler and Brian. And so, yes, it's the results are so compelling. I can see why and how important it is to get through the bureaucracy of the IRB. And with the infusions starting at Mass General, how often will a patient be expected to have the infusions administered?
Yeah, I can answer that. Thank you. That's a good question. And I didn't want to go too much into the actual, as I mentioned on the clinicaltrial.gov website, it kind of goes through that in excruciating detail. But to specifically answer your question, our intent when we manufacture is to get 4 doses of RAPA 501 that are frozen down.
Okay.
A little bit of that depends on the manufacturing, the patient's immune system, but so far we've been able to do that. And the first infusion is performed, and then the second infusion would be about 6 weeks later. And then the next one would be 6 weeks after that, and the next one 6 weeks after that. So about every 42 days, there would be a T-cell infusion, again, as an outpatient, intravenous. Patient goes home the same day. At least that's what's happened so far. There have been no side effects. And other than the infusion and testing, There's no other visits on this EAP. So it's very light relative to the patient having to go into the university. That's the other compelling aspect of the remote monitoring. Allow us to keep tabs on the study participants without them having to travel in to the hospital. Because the cells were very safe, we believe we can do this with only an every 6-week travel back to the trial site.
Excellent. That's wonderful. Thank you.
So the next question is, how will you allocate spots at each center if there are more than 40 patients seek to enroll? And when does these enrolling start and how can we enroll?
Yeah, so the 40 patients, our contract with the NIH is to do those 40 patients over the next 18 to 24 months. So we're on a pretty tight timeline to implement this trial, get it going, which we're meeting all of our milestones, by the way. And so our intent is to treat about 3 or 4 patients every month until we've hit the 40 number. Now, you're right. We do have to try to ensure some equity, especially with these different clinical trial sites, as the investigator and their team exerts a lot of energy to get a study like this up and running. We want to try to make sure that each site that is successful in that endeavor can treat a couple patients at least. So we're going to try to be equitable with respect to that. There will be a rolling admission onto the protocol. So we'll just have to do that assessment as things come to fruition. When we start to look at who's interested in joining the study, we'll do our best to get them to one of the treatment sites, East Coast, West Coast, try to spread it around at least a couple at every site, and to try to be equitable in that regard.
And just to add to that, as Dan said, what we have with NIH under this grant is 40 patients. It's a pretty aggressive timeline, 3 or 4 patients a month from here till the end of next year. Obviously my goal would be to continue the program, right? However we end up doing that, whether it's another grant or, we look to investors or other things. But we're just excited to get this going off the ground, over the next, 3 years. And as Dan said, it's really going to be getting the sites up and running. Every site's going to have, we talked about this yesterday with when we were in Boston with Dr. James Berry. Each site is going to, if the site enrolls, they're going to get, 2, 3, 4 people at a minimum. But we'll be fair. And I would say, I'm an ex CFO. So it's FIFO, first in, first out, right? So, whoever's, we're not gonna handpick one person over another. If they're in the pipe and they're the first in, and then they'll be the next, one first out.
Thank you, Brian and Dr. Fowler. And so another question that we have, you've already touched on Dr. Appel and his work with TREG cell therapy. How is it different? Can you expand a little bit? We've had some questions from what RAPA is, what your treatment is, or is it similar?
Yeah, I would say it's more similar than different. The similarities are they both fit into the Treg family of cells. As I mentioned, Stan's approach is the more traditional approach where you collect the cells coming out of the thymus, kind of the natural Treg, and you expand those up. Ours is a little more crazy scientifically, where you're deprogramming pathogenic cells and you are turning them into a Treg. Now, some of the purists over at NIH, some of my mentors and pioneers say, Dan, those are not Tregs. Oh, that's okay. Well, they have the FOXP3. You saw it going from 0.1 up to 35%. That means it's going to be a good suppressor. So it's a little bit of a epigenetic re-engineering, and it has the hybrid phenotype. So they're very different in terms of how they might work in a particular inflammatory situation. And we just need to do more clinical research to look at how this type of Treg might work versus the one that we've developed.
Yeah.
Now, the other differences are in Stan's research and in many others that have done Treg therapies. You like to give cytokine support for those cells. So you will not only inject the Treg cell, but you will inject a cytokine, an immune stimulation molecule called IL-2, interleukin-2. You'll see that as a combination oftentimes. Treg and cell infusion, and then IL-2 to drive those cells in the body. Our cell, we do not believe that it needs IL-2. Our cells can make their own IL-2. That was in our FDA package, in our preclinical data. So by the merit of our cell making their own IL-2, and by them having this stem cell phenotype, which is not really present in a lot of the other Treg products out there, that our cell will be a more primitive Treg that will be self-sustaining and hopefully be able to exert some beneficial effects for a long time in the body. And that's part of the goals of our research, is to try to figure out how durable these effects might be and how many cell infusions might be the optimal.
Yes.
And let me just to add to that from the, I would say a layperson's perspective, they're both Tregs, as Dan said. The natural Tregs that are in the thymus, trying to find those in the blood, it's like a needle in a haystack. There's not very many of them. It's very hard to expand them because they're already old and senescent. With our process, the way we do it is we're taking the pathogenic cells. They're everywhere. And we're able to convert those to this iTreg, iTreg/Th2 cell. So that's why we've had no manufacturing failures. We can manufacture a lot of this from each patient. Obviously, they have to have some T cells, some level of T cells, as Dr. Fowler said. And then, of course, part of our phase 1 study was to test it without giving it with anything else like IL-2 or any chemotherapies or anything like that. And we were able to do that. So I think, there's a lot of similarities, but I think also there's a lot of differences in what I think people are getting from here is this is a very highly scientific process that we're going through. We're doing it epigenetically, which means we're actually, again, using the natural systems of the cells to reprogram them. And this is a very science heavy project, but with all that, I'm excited because, again, I really want to get this out to as many patients to help them as much as we can, from my own personal experiences with this. So thank you.
Well, I concur. So very exciting. Congratulations. Thank you.
Thank you. I think the next question was, you had mentioned that the criteria for enrollment is less than 50% FVC, SVC. I think there was a little bit of confusion. Did you mean greater than, or is there a minimum FVC and SVC requirement?
Yeah, the protocol is written with slow vital capacity. As you probably know, most people know, those numbers tend to track together, the SVC and the FVC, but we wrote it in terms of SVC. It actually is less than 50%. So it's the reverse of what you typically see on protocols where you're looking for an individual who has 70, 80, 90% of normal so that you don't immediately getting into trouble in terms of breathing. Whereas here, because the definition of expanded access is the patient truly is not eligible for any other trial, we have to go in the other direction and go below 50%.
And so the way I look at it is this is a compassionate use. So, we have our other trials, and the ones we'll be starting next year are like a lot of trials that people have seen where you do have to be at a 70%, or some of these other trials, you have to be at a much higher. So you're getting patients earlier in the disease progression. But this is, again, a compassionate use protocol. It's to give all those other people an opportunity to be a part of this research and a part of this process, and for us to be able to hopefully help them, and again, for us to learn a lot from this.
So the follow-up question was, will there be any other trials if you have not reached SVC less than 50? I think you answered the question on, that would be the trial itself. Thank you.
Yes. And then another question that's coming up is the timing of your phase 2 and 3. Can you talk a little bit about that? Because the expanded access apparently is running alongside that, right?
Yeah, so that is called an expansion cohort. So we were able to work with the FDA to transition from the phase 1 study to a phase 2/3 expansion cohort. And we were able to get that approved also. So that's, as the questioner is stating, running in parallel with the EAP.
Correct. So we originally, the phase 1/2 was at 9 patients. We've now treated 12. So that phase 2/3 expansion cohort is currently, up and running, at Mass General. Plus we have this, and again, our goals is we're working now to design some additional trials that hopefully we can begin next year with even more patients.
Wonderful. Thank you.
So there's a lot of access questions, as you've noticed. Houston Methodist team, they would like to, see EAP at Houston Methodist, and also San Antonio, UT Texas, USC in Los Angeles, and also in Florida. As we see that those are not the centers yet. But the question is, maybe enroll in one of these clinics and fly in for the EAP.
Yeah, I mean, we're not putting any restrictions. I mean, from designing this trial, not putting restrictions on the patients to, obviously they can enroll once a center is up and running. If somebody wants to drive up from San Diego to, LA or wherever they want to fly in, they can obviously contact them. And, our goal is the centers that we showed by the end of the year, then we want to continue to add centers. Adding the centers, it's really getting the centers, getting them to buy off on these things and to want to do that. Cell therapy is very new. So we know even the first patients that we treated at MGH and at Hackensack, people were like, what do? We have to coordinate with our cell therapy center. People have to do an apheresis and give their T cells / blood. It's actually fairly simple, but I think just when people hear that, or even investigators hear that at certain places, it seems very scary to them because they've never done it. It's very new. So if you have a certain center that you really like, I would say pressure your doctor to, contact us and join the study. Because whether it's this study or the next one, our goal is to get it out to as many as possible. But there's no restrictions on people signing up and flying or driving to a site that's active.
Perfect.
Thank you, Brian. And you're pretty much answering a lot of the next couple of questions. So we have a lot of people on the call tonight asking just what you were talking about, the logistics. So we have someone who's a patient at UCSF in San Francisco, and they're basically asking, should we contact or should they contact their neurologist to participate? And it sounds like you're answering that. And the same at MGH. And I guess the other question is, as Indu was mentioning, Florida seems to be, I'm sure you're working with places in Florida, but what about Nova? Is there a possibility of them being a study site?
I mean, I think anything's a possibility. Again, we're pretty regimented. So we have like I said, we were at up in Boston yesterday with James Barry. We're actually we're leaving to get to Hackensack's. We'll be there all day tomorrow. So we're actually, hopefully we don't miss our planes. So, we are going to be moving through the centers that we showed from here to the end of the year, but I always say anything's possible. The more centers we can get on. So if somebody's interested, they'll call. Again, I'm an ex-accountant. It's first in, first out. These centers, we're already working with them, but we'll continue to want to add as many as we can, next year and the following year, and then for our other studies that will be coming down the pike.
Thank you.
Thank you so much. And I think we have one last question. There are people from non-ALS denominations such as PLS, HSP. Sometimes they come to the community because they don't have certain, similar community. So would this actually help HSP as well? Do you have any thoughts about how it can actually help expanded, ALS cousins, as they call it?
Yeah, that's a good question. We believe that our RAPA 501 cell is going to be suitable for many inflammatory conditions, whether you're talking about ALS or ALS variants, Parkinson's disease or lupus, and so on and so forth. So that's what we think is the potential of the treatment because of the way it's designed and how it works. So it should be relatively broadly applicable, but obviously that's easy to say and difficult to prove and to get the resource to expand it out to other clinical scenarios. But that would be our dream, to be able to expand both within ALS and into other conditions.
Thank you. One last question, Dan, that I wanted to make sure that we address this. Can the participants be in other trials after the infusions of RAPA?
Can you clarify the question? I'll turn it back.
I think there's a lot of questions on you can only be in one trial at a time, or can they actually get into EAP? And if they're actually eligible for another trial, is it okay to get into that trial post infusions?
Yeah, I don't think we really address that within the protocol. Classically, if it's an intervention trial where you're getting another experimental drug, it never happens where you're on this study and that study.
Yeah.
So it's probably I would say not the right thing to do for either study. More importantly, for the second study, it would be important that they come off of our study.
And I think just to add from a practicality standpoint, I think once they're done with the infusions and they've gone the to the end and we've measured everything over a 6-month, 8-month, 9-month period, then if they wanted to, come off of our study and go on something else, that's great. But as Dan said, we do want to answer a lot of research questions here as to, kind of the data that people saw where the slow vital capacity stabilized. That's because we were able to give them the therapy and measure it for 6 months, right? So we would not be able to answer those questions, but there's nothing hindering them from after infusions and they come off of ours to go on anything else.
Thank you. And we don't want you to miss your flight. And it's very rare we actually, go almost an hour and a half. So thank you very much for your time. And we can't thank you enough. And anybody who have questions, I think we have put Dan's email address and they can contact us at, everythingals.org, and we'll pass it on to Dan and Brian. So thank you very much, Dan and Brian, and for everything you guys do, really, and we look forward to having this a success working with you.
Thank you all. We really appreciate everyone here.
Thank you.
Thank you for all your support.
Yes,
appreciate it.
This is your extended family, and we do love you because, to me, you all make a big difference in each person here. Because, see, a lot of you don't know each other, but you know that you have the same goal in mind, just like all of us. So, if no one else has a comment, I want to make one more appeal, and that is for everybody to pick somebody here who you see tonight and speak their name and say goodnight to them. And let's do it on 3. 1, 2, 3.
Good night, Sherry.
Good night.
Good night.
And in 2 weeks, we're going to bring in another company called Prolinia, who's going to talk about their EAP program. So we'll see you soon.
Guys, thanks, Finn. Thanks, Dee.
Good luck, Linda.
Good night, Mario.
Night.
Good night, Tracy.
Good night, everyone. See you next time.
See you next time, Chuck Shaw, Brian Kelly. Hansel, thank you for being here. Paul Atkin. Paul, good night, Paul.
Bye,
thank
Dee Dee.
you so much for joining us.
Everybody do the heart.
That's right.
Yeah, keep practicing. I'm
Each and every one
getting
of
a
you.
little better, but I'm watching you all. I'm trying to figure you out. Love it. Mike McHenry. Mike, good night. Thank you for joining us today, brother. And Tansel, that's right. That's all I wanted. A little smile and a wave. And Philip Weiser, Philip, thank you for bringing the Golden Gate Bridge with you tonight. Everybody brings something special. And with that, we're going to call it another day, folks, and we'll see you in 7. All right? All right. Yes, Ted.
Thanks, McFinn.
Thank you, Ted. All right. Good night, everyone.
Good night.
Thank you. Good night.